Bismillah ar-Rahman ar-Rahim. Now we are going to start one of specialized connective tissue in our body, and blood. Blood is a specialized connective tissue with a fluid extracellular matrix, and it's make about 7 to 8% of our body weight. And the average adult person have a 5 to 6 liter blood in the vasculature, in the circulatory systems. The blood out of the vasculature, out of the circulatory systems, the protein in the blood interact with each other and produce a blood clot. And over this blood clot we have somehow yellow liquid, viscous liquid called serum. Actually the serum is a plasma with more growth factor minus clotting factor, because this clotting factor participate in clot formations. About the function of the blood, you know the blood have a transportation function. Actually the blood distribute the gas, oxygen and CO2 in our body, nutrient and waste materials, and hormones. The blood have a regulation function, regulate the pH, temperature, and also the osmotic balance by transferring water to and from the tissue. Blood with having leukocyte or white blood cells having antibody and other protein that they can destroy any invaders, because having of these component, so the blood has protection roles in our body. Again with anticoagulant factors have another protective functions and prevent bleeding in our body. But when we add anticoagulant factors or anti-clotting factors to the bloods, after centrifuge, by this way actually we prevent the clotting. And after centrifuging the blood, we can see 3 layers in sample tubes. On the bottom of the tube, the sedimented material in this area called, actually they are a erythrocyte or red blood cell. The volume of the erythrocyte called hematocrit. In the male about 40 to 50% of blood volume, and in the female you can see somehow below 35 to 45% of blood volume. Over this area we have a liquid or viscous liquid area, and also it's a transparent, translucent called the plasma. The plasma make 55% of blood volume. 92% of plasma is water, 70% of them is protein, and 1% belong to the electrolyte, nutrient, respiratory gas, and the waste and products. Below the plasma and between the plasma and also hematocrit, we have a gray-white sedimentation or area or components called buffy coat. The buffy coat composed of the platelets and white blood cell or leukocyte. About this area makes about 1% of blood volume. Let's start with the plasma protein. We have 4 types of protein in the plasma. The first one is albumin. Albumin is most abundant protein in the plasma. They maintain the osmotic pressure in the vasculature, and also it's help us to carry off the phobic materials like fatty acid, like unconjugated bilirubins, and these materials transport within the blood by albumin. Another protein in the blood is a fibrinogen. The fibrinogen is the largest plasma protein, sometimes the largest plasma protein. And after or during the blood clotting, this protein polymerized and make a network shaped structure called fibrin, and this fibrin block the site of bleeding in the vasculature. About the complement proteins, I will explain the next slide. Let's go to the globulin, because they have a globular shape, spherical shape. We have two types of these protein, alpha, beta, and gamma. Usually the alpha and beta globulins, they are a transferring protein. For example, one of them transfer the iron called transferrin, or other proteins, or some of them transfer the lipid called lipoprotein. Others, they don't have a transferring function, like a fibronectin. Here we have a soluble fibronectins, and prothrombin function as a anticoagulant, function as a coagulant factor in the blood. And the third one, the gamma globulin, is actually antibody. Is another name is immunoglobulin. Function against any antigen. All of these proteins are synthesized in the liver, except gamma globulin. This protein produced by a plasma cell in the connective tissue. About the complement proteins, we have about 30 types of these protein in our blood, and when and the antigen of the pathogen recognized by an antibody, these reactions actually activated one members of these superfamily proteins. For example, C1, we call them C1, C2, C3 in this manner. The antibody is enough to activate one of them, and then the activated component start to activate other component in a cascade manner, and finally they assemble and make a pore on the surface of a pathogen, for example in the microbe, and cause a pathogen die. Here you can see again, antigen is recognized by antibody. Then antibody activated, for example, C1, complement protein 1, and this protein in a cascade manner you can see activated the other proteins, and finally the assembling some of them make a pore on the membrane of any invaders and microorganisms, and these pores cause swelling, and at the end we have we can see cell burst and cell dies. For seeing the blood under the microscope, we don't need any tissue sectioning or need tissue cutting by a microtome. We just need a drop of blood on a slide, then spreading into a thin layer by another slide. After fixation, we can stain it with some special dye. Usually this kind of dye is a mixture of the acidic, basic, and sometimes the acidophilic dyes under the name of some scientists, for example, Gimso, Wright. the name of scientists. You can see the mixture of, for example, the methylene blue basic dye, eosine acidic dye, and acero B acidophilic dye. And this mixture of dye can stain all component, all cellular component within the blood, so we can easily see under the microscope. For example, here you can see a microscopic slide. We put a drop here and spreading it on a slide, and this spreading gives us three area, and the proximal, distal, and middle part. For better seeing the cells, the middle part is very good. You can see regular and also the event distribution of the cells, but in this area we can see a few cells, or this one we have a compact and multi layers of cell, but the middle part is very good to seeing the cells. This is, you can see one of these slides stained with a Gimsa dye. We can recognize the red blood cell and white blood cell and platelets. Let's go to the knowing or understanding the property of the blood cell. In the blood we have monocyte, lymphocyte, neutrophil, eosinophil, basophil, and erythrocyte or red blood cell, and platelets. Start with erythrocyte. Erythrocyte, you know, is a biconcave cell. These cells lack nuclei and organelles, and the cytoplasm filled with the oxygen carrying protein hemoglobin. The diameter of the cell, you can see about 7.5 micrometers, usually as a standard size within our tissue under the microscope. Smaller than this size called microcyte. Bigger than this size, about bigger than 9 micrometer, called macrocyte. Number of the density of red blood cell or erythrocyte is about, in the male, is about 4.126 millions in micrometers, microliters, and in the female 3.9 to 5.5 millions in microliters. The red blood cell are a flexible cell. They can change the shape to passing through the narrow vasculature, for example, through the capillary, and they are so flexible to passing this pathway. And in the large blood vessel, actually some of them attached together in this way and make a stack of cell. This structure called rulex, like this. This attaching is very loose and easily they can detach from each other. The membrane, about the membrane of RBC, you know, most of the information, our information about the cell membranes comes from the information of the red blood cell membrane, because these cells doesn't have any organelles. So all of the membrane we catch from this cell is belong to the cell membrane. And another important factor is this cell is very accessible, or we can easily take a sample of these cells from anyone, and to obtain the cell membrane, to study the cell membrane properties. For example, we know the red blood cells, we have about 40% protein, the 50% protein, 40% lipid, and 10% carbohydrate. And the protein in a red blood cell membrane we have two kind of protein, transmembrane protein or integral proteins and peripheral proteins. The important integral proteins like band 3 and glycophorin A, usually they function as ionic channels. And important peripheral protein like spectrin and ankyrin, they are very important in maintain a biconcave shape of red blood cells. By this way, they can increase the surface to volume ratio, so they facilitate the gas exchange between the oxygen and the environment, between the erythrocyte and or we can truly say between the hemoglobin and the environment. And also this protein spectrin ankyrin maintain the cell elasticity or the flexibility. Without this protein, the cell get spherical and we don't have a biconcave shape. Usually the carbohydrate in the cell membrane attached to the protein from the outside, not from the inside cytoplasmic area. And this carbohydrate you can see here usually make a ABO typing blood groups. I'm sure you know them. For example, in a blood type A, we have a carbohydrate type A. In blood type B, the carbohydrate change. And in ABO, we have both of them. And in the type O, we don't have them. And everyone has blood type A. They have antibody against type B. And vice versa. So type B have antibody against type A. So we cannot transfer the blood between this kind of person. ABO, because have both of them, they don't have any antibody against these blood type groups. And in the O types, we have antibody against both of them. The question here, as an assignment for you, is why we have antibody against, for example, in this person, we have against B. But this person never get a blood from this person to producing the antibody. Here you can see the carbohydrate on the surface of red blood cells. They are very similar, just they have differences within hexane sugar with 6 carbon atoms. We don't have here this sugar or monosaccharide, and in this one type B, you can see we have different types of sugar in this part, but the other sugars are very similar. So in different kind of blood types, we just have a minimal differences in the carbohydrates. The cell, the RBC, lose all of the organelles and nuclei and fill the cytoplasm by a oxygen-carrying protein called hemoglobin. Hemoglobin is made of 4 change of polypeptides, and each change have a heme group, and also heme have a iron component. By this iron component, they can attach to the oxygen and CO2 in a reversible reaction. So when the hemoglobin attached to the oxygen, make a oxyhemoglobin. By attaching to the CO2, make a carbaminohemoglobin. Both of them are reversible reactions. It means they can easily detach from each other. But when the hemoglobin attached to the CO monoxide carbon, it's an irreversible reaction, and we cannot easily detach them, and we need a high concentration of oxygen to detach them. Because the RBC, red blood cells, don't have any organelles and nuclei to replace the proteins and other components in the cells, and all of the components of proteins or lipids in the cell membrane, and they have a limit lifespans and need to be replaced by a new one. But in this cell, we don't have any organelle to produce new one. After 120 days lifespan of the RBC, we have a defected membrane because of this kind of damaged protein on the cell membrane. Cell swells or swell up, and some of the proteins damage on the cell membrane. All of these changes in the cell membrane recognized by a macrophage, especially in the spleen, sometimes in the liver, and also the bone marrow. The macrophage in these places recognize the old RBC or senescent RBC, phagocyte them, and degraded them by a lysosome enzyme. The cell filled by a hemoglobin, so the hemoglobin has two parts, heme and globin. Heme has again two parts, porphyrin and iron. The iron detached from the heme and stored within the cell in a condensed shape called ferritin or hemosiderin. Porphyrin as a pigment converted to the unconjugated bilirubin, transferred to the blood. From the blood, within the blood you can see carried by albumin, then come to the liver, absorbed by a hepatocyte, then hepatocyte converted unconjugated bilirubin to the conjugated bilirubin. Now the digestive system and also the urinary system can excrete this kind of poison materials. And globin is a protein, so degraded to amino acid, the cell can again use this amino acid. Some disorder about the blood, hemolysis is the rupturing or breaking down of the RBC and releasing of hemoglobin into the blood, called hemolysis. When we have a increased rupturing of RBC, the liver cannot convert the unconjugated bilirubin to the conjugated bilirubin, so remain in the blood and create jaundice or icterus. You can see the yellow skin because of increasing unconjugated bilirubin in the blood. Anemia, when we have a low concentration of RBC compared to normal range, they have symptoms of or they can be this kind of person can be characterized by fatigue, heart palpitation, shortness of breathing, pale skin. These are the symptoms of anemia. And polycythemia, when we have a high concentration of RBC, so they increase the blood viscosity. Sometimes in some people can cause of a stroke, brain stroke, or sometimes kidney stroke because of high concentration. They block microvasculature. Sometimes this polycythemia is physiologic, especially the people live in a high altitude for a long time, and this kind of person, in order to compensate low tension of oxygen. They produce a lot of RBC to compensate these hypoxia situations. So they have a physiologic polycythemia. And the last one, sickle cell anemia, is because of mutation in the DNA. You can see the shape of this kind of red blood cell is different from the normal. The scientists say they are a side shape or cycle shape. After centrifuging the normal blood, you can compare it with the anemia or polycythemia. In the anemia, you can see the volume of hematocrit decreased. In the polycythemia, you can see the volume of hematocrit increased. In the sickle cell anemia, we said we have a mutation in the DNA. This causes amino acid substitutions. For example, instead of the glutamic acid, we have a valine. This creates a cycle hemoglobin. In the cycle hemoglobin, after the oxygenated, these hemoglobins polymers attach together and make a fibrins, and these fibrins within the cytoplasm change the shape of cell. Here you can see normal hemoglobin. After they are oxygenated, they are single and distributed in between the cytoplasm. But in the sickle cell anemia, you can see they attach them together and make a fibrous structure. And these fibrous structures cause protrusion or extension of the cell membrane. You can see this fiber within the cell cytoplasm. And these fibers, in addition to changing the shape of the cells, decrease the cell lifespans, also decrease the cell flexibility, so they cannot pass through the narrow vasculature like a capillary. Sometimes they block this blood vessel and cause the stroke. Here again you can see normal hemoglobin distributed singly within the cytoplasm, but in the hemoglobin type of cycle or hemoglobin S, you can see they make a fibrous within the cell and change the shape of cell. Again here you can see the fibrous polymerization of cycle hemoglobin. Here I have a cartoon. I don't know you see this cartoon. The name of this cartoon was Once Upon a Time Life. You can see RBC, they are carrying oxygen. It was very interesting cartoon during my teen ages. Let's go to the understanding another groups of cells within the blood, of white blood cells or leukocyte. Leuko means white, cyte means cell, again white blood cell. Monocyte, eosinophil, basophil, lymphocyte, and neutrophil. We can classify the leukocyte in two groups according to having granules or without granules. Granulocyte, agranulocyte. You can see in the cytoplasm of these cells we have a granule, but these blood cell, a granulocyte don't have this type of granules. Another classification according to the shape of the nucleus. Some of them have a spherical, some of them have a spherical with a indented. This kind of leukocyte called mononuclear, and some of them have a lobulated, segmented nucleus. But all of these segments of lobes attached together by extension of a nuclear, so we cannot call them multinuclear. Actually they have one nucleus but in a different shape with different lobes. so-called polymorphonuclear. With both of these classification, we have a neutrophil, eosinophil, basophil in one groups, and lymphocyte and monocyte in another groups. The granulocytes like basophil, eosinophil, neutrophil. The polymorphic nuclear leukocyte, we can again say neutrophil, eosinophil, and basophil. And a granulocyte like a lymphocyte, monocyte, mononuclear, again lymphocyte, monocyte. Number of the leukocyte in our blood is about 4,500 to 11,000 in microliters. The leukocyte, unlike the RBC, they do their function in the outside of vasculature, in outside of blood vessels. So they should pass through the wall of blood vessels and go to the extracellular matrix to play their role. But for this function, actually start, for example, from here, we have an inflammation area, inflammation site. The cell in the inflammation site, for example, the macrophage in this area, produce some cytokine like a tumor necrosis factor, interleukin-1. This is called cytokine. This cytokine affected the endothelial cells of blood vessels, the epithelium of blood vessels. They loosen the intercellular junctions in this area. In addition, they bring on the surface one type of receptor called P-selectin and E-selectin. The leukocytes have a ligand on their surface for this receptor, so they attach them and speed down. And after that, they make a firm junction by producing another groups of protein called integrins. And by the integrins, they can stop completely. After that, they make some extension to outside of the blood vasculatures and migrate to outside and head to the site of inflammation to kill and protect our body against any invaders. This process called diapedesis. Diapedesis means jumping through the blood vessel wall. The P-selectin, when they during this diapedesis appearing on the cell membrane, actually we have this protein, cell membrane protein, within the granules called Weibel-Palade bodies within the endothelial cells. After stimulation, we have exocytosis of these granules, and after exocytosis, we have appearing of these protein on the cell surface. Granulocyte, they have two kind of granules within the cytoplasm. One group specific granules, another group azurophilic granules. The another name for a specific granules called secondary granules. For the azurophilic granule called primary granules, or another lysosome. You know, most of the cell have a lysosome. So these kind of granules we have in other groups of cells, even in granulocytes. But the granulocyte cells, in addition to the lysosome, they have another groups of granules called specific granules. These specific granules can be stained by a acetic dye or basic dye or neutral dye. According to this staining, we can classify the granulocyte in 3 groups of cells: neutrophil, stained with a neutral dye; eosinophil, stained with a acidic dye; and basophil, stained with a basic dye.